Hi, this is Ferdinando Almeida here at the Stein Monogastric Nutrition Laboratory, and I'm here today to discuss with you the results from two experiments in which we measured the amino acid digestibility and concentration of digestible and metabolizable energy in a threonine co-product fed to weanling pigs. This is the outline of this presentation. I'll first give you a brief introduction, followed by experiment 1 and 2, discussing some of the materials and methods and results, and I'll finally leave you with overall conclusions. Success in feeding weanling pigs is dependent on the selection of high-quality feed ingredients such as fish meal, which has been proven to be a good animal protein source due to its highly digestible crude protein and amino acid levels. However, the rising cost of fish meal in the past few years has contributed to a continuous evaluation of potential protein and energy sources that can replace specialty feed ingredients such as fish meal. Synthetic threonine has been supplemented to low crude protein diets to improve the nutritional value of these diets and also to meet the requirements of threonine for these animals. Because of that, the commercial production of synthetic threonine has continuously increased, and that's mainly due to the developments in large-scale fermentation technologies. In this slide and the next one, I'm going to try to briefly show you how the production of synthetic threonine occurs. And so the very first step, what we have is a fermentation broth that contains, for example, a bacteria, E. coli, and a carbon source. So in this case, we have glucose, and glucose can be converted to oxaloacetate by this bacteria, and it's also further converted to aspartate. In a second series of reaction, aspartate is converted to aspartate semialdehyde, to homoserine, and finally to threonine. So that's the first step in this production of synthetic threonine. Now let's move on to the following steps, and we'll then describe what is the threonine coproduct that we are using for these two experiments. After the fermentation broth steps, if we look at the left side of this slide, we see that there's a cell separation step followed by a pH adjustment, crystallization, separation, drying of the product, and finally, on the bottom on the right side, we have the synthetic threonine. However, on the cell separation step, there is a biomass product that is left, and that biomass can be further processed into the threonine coproduct, which is the product that we used in both experiments that we'll be discussing here today. This slide shows a picture of the threonine coproduct and just showing a little bit of the physical characteristics, which is a flaky type of ingredient which contains dry matter of approximately 95%, crude protein approximately 81%, 8.4% of fat, and also contains approximately 5,800 kcal per kilogram of dry matter of gross energy. This graph shows the amino acid composition of the threonine coproduct compared with fish meal. So on the y-axis we have the percentage of amino acid. On the x-axis we have just the first four limiting amino acids: lysine, methionine, threonine, and tryptophan. And for all amino acids, for all these four amino acids, we can see that the threonine coproduct contain greater levels of lysine, methionine, threonine, and tryptophan compared with fish meal. I want to point out that the threonine coproduct contain almost double the amount of threonine compared with fish meal, 4.5% in the threonine coproduct versus 2.4% in fish meal. And the reason for that may be that during the cell separation process, in the steps of production of the threonine coproduct, maybe not all the threonine that was produced in the fermentation broth step is removed from the cell separation step, and therefore we may end up with greater concentration of threonine in this threonine coproduct. To this point, no experiments have been conducted to evaluate the nutritional value of the threonine coproduct, And therefore, our objectives here were first to determine the apparent and standardized digestibility of amino acid in threonine coproduct and fish meal, and also to determine the concentration of both digestible and metabolizable energy in corn, threonine coproduct, and fish meal, and to compare these values among these ingredients. So for experiment number 1, we had 9 wheeling barrels with an initial body weight of 13.4 kilograms, and they were equipped with a thick cannula in the distal ileum. They were allotted in a triplicated 3x3 Latin square with 3 diets. The first diet had 20% inclusion of the threonine coproduct, and therefore we had a diet containing 18% of crude protein. The second diet contained 25% of fish meal, and the diet contained also 19% of crude protein. And the third diet was a nitrogen-free diet that was used to measure the basal endogenous losses of amino acids and crude protein. And we had 3 periods for these experiments. For the statistical analysis, we used the mixed procedure of SAS. The model included diet as a fixed effect, replicate as a random effect, and we used the LS means option to calculate the means for each treatment. We used the pdiff option to determine significant differences among the means and an alpha level of 0.05. Now let's take a look at the results. All the graphs in the results part, they follow the same pattern, so I'll just take a moment here to explain the pattern. On the y-axis we have digestibility in percent. On the x-axis we have on the left apparent, and on the right standardized digestibility. And these first results here is for the apparent and standardized digestibility of lysine, and we can see that threonine coproduct had greater apparent and standardized digestibility compared with fish meal, 87.2% for the AID versus 81.2% in fish meal, and 92.7% SID in the threonine coproduct versus 85.3% for fish meal. The second graph shows the results for methionine, and here once again we observed that threonine coproduct had greater apparent and standardized digestibility of methionine compared with fish meal. These are the results for threonine. The threonine coproduct had 88.4% AID compared with 66.1% in fish meal. And for SID, we had a digestibility of 93.9% in the threonine coproduct versus 76.8% in the fish meal. Now this graph shows the mean SID for indispensable amino acids on the left, dispensable amino acids in the center, and all amino acids on the right. So we observe here that threonine coproduct had greater SID of indispensable, dispensable, and also mean of all amino acids compared with fish meal. If we take the results from this experiment to calculate the concentration of SID amino acid in threonine coproduct and fish meal, we have these numbers shown in this table. So for lysine, threonine coproduct would contain 4.82 grams per kilogram compared with 4.06 in fish meal. For methionine, the threonine coproduct would have 1.98 grams per kilogram compared with 1.45 in fish meal. For threonine, we would have 4.25 grams per kilogram in the threonine coproduct compared with 1.84 grams per kilogram in fish meal. And for tryptophan, we would have 0.94 grams per kilogram in the threonine coproduct versus 0.53 grams per kilogram in fish meal. So it's consistent here that the threonine coproduct would contain greater SID of at least the first 4 limiting amino acids compared with fish meal. So in conclusion from this first experiment, the threonine co-product had greater AID and SID of amino acid compared with fish meal. Now let's look at experiment 2, and in this experiment our objectives were to measure the digestible and metabolizable energy in the threonine co-product compared with digestible and metabolizable energy in corn or in fish meal. For this experiment, we used 24 barrels with an initial body weight of 18.1 kilograms, and they were placed in metabolism cages and allotted to a randomized complete block design with 3 diets. The first diet was a corn basal diet containing 96.4% of corn. The second diet was also based on corn with 79.3% corn and 17% of the threonine co-product. So in this diet we had approximately 4,000 kcal per kilogram of gross energy and 21.2% crude protein. And the third diet we had 75.3% corn plus 25% of fish meal, and this diet contained also approximately 4,000 kcal per kilogram of gross energy and 21.4% of crude protein. And we had 8 replicates for each diet. The pigs were allowed for a 5-day adaptation to the diet and to the crates, followed by 5 days of total collection of feces and urines. And the statistical analysis that we used for this experiment were the same as described for experiment 1. Now let's take a look at the results. This first graph shows the DE concentration, and we have, so on the y-axis, DE in kcal per kilogram of dry matter. The orange bar again is threonine co-product, the blue fish meal, and yellow represents corn. And these results show that threonine co-product had greater concentration of digestible energy compared with both fish meal and corn. And fish meal also had greater concentration of digestible energy compared with corn. The results for ME concentration show that threonine co-product was not different from fish meal. However, both threonine co-product and fish meal contained more metabolizable energy compared with corn. Therefore, the overall conclusions from these 2 experiments were that the threonine co-product has been shown to be an excellent source of crude protein and indispensable amino acids, and also it's a product that contains high concentration of digestible and metabolizable energy, which are greater than in corn, and in the case of ME is similar to fish meal. Therefore, this is a high-value alternative feed ingredient for weanling pigs that can potentially replace either partially or totally the fish meal for starter diets. With that, I would like to thank ADM for funding our research. And I would like to thank you for listening to this podcast. You may visit our website at nutrition.nsi.illinois.edu and find out more about this ingredient and others that our lab conduct research on.